But if you could just start the live stream to YouTube. Once that's up, our on-air lights will come on in the space. I'll do a very brief introduction from over here, and then I'm going to pass it over to Jordan to take it away from there. So just hold on, everyone. Also, thanks so much for joining us, by the way. This is really fun. Hello, everyone. Welcome to Concordia University's Fourth Space. Thank you for joining us for today's event, 3-Minute Thesis. Now, this is the Northeastern American Association of Graduate Schools Regional 3MT competition. We would like to begin by acknowledging that Concordia University is located on unceded Indigenous lands in Tiohtià:ke, Montreal, and we respect the continued connections with the past, the present, and the future in our ongoing relationships with Indigenous and other peoples within the Montreal community. Here at Fourth Space, we work with our university community to help mobilize knowledge. We co-create daily activities examining research questions, projects, things happening all across our university. Now, we're sharing this competition as a live stream to YouTube, as well as a Zoom meeting. So if you're joining us via Zoom, comments or questions, pop those into the chat. We're gonna try and do a little Q&A a little bit later on. As well, you can feel free to use the reactions button on Zoom. We can see that here in the space as well. And so as well, for those of you here joining us in the space, if you have comments or questions later on, raise your hand and we'll run a microphone around to you so that everyone can hear. Finally, we will be running an online survey for the People's Choice Awards at the end of the competition. There will be a QR code visible via Zoom, and we'll add the link to that also well in the chat. Those of you here with us in the space, there's a QR code there as well, you'll see. Okay, with that, it's very much my pleasure now to pass the microphone over to our Associate Dean, Graduate Academic Programs and Development here at Concordia University, Dr. Jordan LaBelle. So great to have you in.
Thank you so much. Good afternoon, everyone, and welcome, welcome, welcome. It is my pleasure to host and welcome you to the 2026 Northeastern Association of Graduate Schools 3-Minute Thesis Competition, hosted by Concordia University right here in beautiful Montreal with the support of our dear and long-term partner, Fourth Space. The entire team here is just so wonderful and so welcoming and work their magic to handle all the back-end logistics to make every event they host a smashing success. So a big heartfelt thank you to them. We are delighted to welcome participants, judges, colleagues, and guests joining us from across the Northeastern region today and even beyond. So thank you all for joining us, for lending your own passion, enthusiasm, and energy to make this what I know will be a memorable and uplifting event. If you've attended any 3MT presentation, you know what I'm talking about. You tend to go out of these events with a bounce in your step and renewed hope for the state of research and, dare I say, the future of the world. This year's competition began with a very strong first round. We received submissions from 46 institutions, representing 11 US states and 2 Canadian provinces. We also saw an impressive diversity of degrees being represented across disciplines in this competition, including graduate research in engineering, biological, biomedical sciences, health, earth and environmental sciences, public health, education, nursing, psychology, chemistry, sustainability, even the arts, and I'm probably forgetting a few, and I apologize if I did. Today's event brings together the finalists of this year's Northeastern Association of Graduate Schools 3-Minute Thesis competition. These presenters have already distinguished themselves through the clarity, the creativity, and the impact of their research communication, and we are very pleased to have them with us today for this regional final. It is now my pleasure to invite the one and only, my boss, Dr. Jeff Dover, Interim Dean of the School of Graduate Studies at Concordia University, to offer some opening remarks.
Thank you very much, Jordan. Thank you. So good afternoon. Welcome, everybody, to the 2026 3-Minute Thesis Competition. I'm Jeff Dover, the Interim Dean of Graduate Studies here at Concordia, and it's a real pleasure to welcome everyone here today and online, or wherever you're from. The 3-Minute Thesis Competition is one of those rare academic events that manages to both be impressive and slightly terrifying at the same time. Whether the challenge is simple in theory, to just explain your research, the essence of your research in 3 minutes, but it's really challenging. And they can only use one slide, and this means that there's no long introductions, no detailed literature review, and no comforting promise that the really important part's coming in about 10 minutes, because you've only got 3 minutes to do your presentation. So, and this is exactly what makes the competition so valuable. It gives students the opportunity to refine one of the most important skills they can develop as researchers, which is the ability to communicate clearly, confidently, and meaningfully beyond their own discipline. Research does not only live in dissertations, journals, or conference rooms, it also matters in the wider world, and being able to share that work in accessible ways is an essential part of scholarly life. Today's event features top graduate students presenters from the NAGS, which is the Northeastern Association of Graduate Schools, all of whom have already distinguished themselves in their local competitions, and reaching this stage is already a significant achievement, and it speaks not only to the quality of their research, but also the thought, preparation, and care they've put into communicating it. I've been involved with a 3-minute thesis at various levels across different universities, and it's always great to see the advancement of students, seeing how the presentations change over time, and you guys are really in for a treat today, because getting to this stage, these presentations are really buttoned up to where they were months ago and just practicing them. So I'm really looking forward to seeing them. On a personal note, I have been in academia for most of my life, and I'm continuing to be struck by the graduate students' creativity and commitment and their ability to rise to challenges like this one. Competitions such as the 3-minute thesis remind us that graduate students are not only doing excellent and important research, but they're also finding ways to make that work understandable, engaging, and relevant to others. I would also like to thank the volunteer judges for contributing their time and expertise, and to thank everyone involved for organizing today's event. These competitions depend on a great deal of work behind the scenes, and we're grateful all for it. To all of today's participants, congratulations on everything you have achieved to this point. We're very pleased to have you here, and I wish you all the best in today's competition. Thank you.
Thank you very much, Jeff, for those insightful and thoughtful remarks. I noted the words impressive, but slightly terrifying. Yes, it's true. Anybody who had to take on this task of summarizing their research and for at least 3 minutes, quieting those little voices in our head that tend to cast doubt perhaps on our own abilities, know what it's like to tackle that demon. That's part of what makes this competition really so exciting. We are not only celebrating excellent graduate research, but we're also celebrating and elevating the skill, the judgment, the creativity, the unique competencies that it takes to communicate that research clearly, memorably, and in a compelling way to an audience of people who don't know your particular field, don't know your particular topic. And as we all know, that's probably harder to do than it sounds, right? A few important rules, Jeff touched on some of them. Each participant is allowed only one static PowerPoint slide, so no animation, no transition, no props, no audio, no video are permitted. Presentations must be delivered as spoken word, and the timing will begin when the presenter starts, either through speech or movement. And then finally, any presentation that exceeds 3 minutes will be automatically eliminated or disqualified. Our judges will evaluate presentations based on communication, comprehension, engagement, and also how effectively the slide supports the presentation. Before we begin, I would like to extend a special thank you to our judges for lending their time, their expertise, their passion for this competition. Amy Cison is the Associate Vice President and Dean of Admissions at Wentworth University of Technology. Yvonne Lee is the Director of Writing across the curriculum at Lehigh University. And Jovana Milosavljevic Ardelian, is the Graduate Education and 3MT Program Lead at the University of New Hampshire. And Jovana, I am so almost jealous and admiring that your university has a program lead for the 3-minute thesis. I think it speaks to the importance and value your organization puts on this event and this competition. So special thanks to Amy, Yvonne, and Jovana for supporting this competition with your evaluation of today's presentation. I would also like to acknowledge the contribution and help of Ramazan Gongor, who is the Assistant Dean for Professional Development at Rutgers University. Ramazan helped to judge a previous portion of this competition by watching all the videos. Unfortunately, he could not be with us to judge today's live presentation, but a heartfelt thank you to him as well. Today we have 15 participants divided into 2 groups with a short break in between. And we'll all need that break, our participants and ourselves as well, to sort of digest all that wonderful work and inspiring knowledge we'll be exposed to. We are also pleased to have a People's Choice Award in this competition that's selected by you, the audience members. Voting for the People's Choice Award will open precisely at 3:35 and close promptly at 3:45. So I urge you, you can do anything in between, but be in front of your screen, be glued to your screen from 3:35 to 3:45 if you want to vote for the People's Choice Award. Audience members will be invited to vote using the link in the chat box or the QR code that will appear on screen. After the judges deliberate, we will announce the winners of today's competition. And those winners will have cash prizes. We will have 2 co-winners who will be awarded $400 each. The People's Choice Award will receive $300, and the co-winners will advance to the National 3-Minute Thesis competition hosted by the Council of Graduate Schools during their annual meeting next December. So thank you all for being here and for helping make this competition possible. Before we get going, please allow me to use my prerogative as host to extend my heartfelt encouragement and best wishes to all the participants. Take a deep breath. You've done this before. You've done the hard work. You've done the hard press. Now enjoy the process and let your superpowers shine. So now let's begin. Our first presenter is Janelle Harding, who is completing her Doctorate of Nursing Practice at Westchester University. The title of her presentation is When Seconds Matter, Redesigning Sepsis Care. Take it away, Janelle.
Imagine having what you thought was a simple infection, but then your fever gets worse, your heart begins to race, and you feel weaker as the hours go by. So you decide to go to the emergency room. Within hours, your blood pressure drops, your organs begin to fail, and you're told you have sepsis. Sepsis is the body's overwhelming and life-threatening response to an infection. It progresses quickly, and every hour in delay of treatment increases your risk for death. In the United States alone, sepsis costs nearly $62 billion annually, and 1 in 3 patients diagnosed with sepsis do not survive. But here's the important part. We already know how to treat sepsis. If clinicians rapidly administer IV fluids, antibiotics, and collect critical labs, survival improves dramatically. And to ensure that this happens, the Centers for Medicare and Medicaid Services created a national quality measure called SEP1, requiring hospitals to complete these steps within strict timeframes. But SEP1 is an all or nothing measure, meaning if any one of those steps is delayed or missed, the hospital is considered non-compliant. And nationally, hospitals succeed 60% of the time. And this isn't because clinicians can't recognize sepsis. It's because the system around them is fast, complex, and unpredictable. Even so, sepsis can look like heart failure, sepsis can look like pneumonia, and sepsis can look like nothing until it's too late. So I asked the question, what if the system could do better? In modern hospitals, healthcare delivery is done through the electronic health record. If a clinician suspects sepsis, they would use a sepsis order set, a pre-built group of orders that would include those IV fluids, antibiotics, labs, and monitoring. But order sets are often long, complicated, and not aligned with clinical workflow. In a 500-bed level 1 trauma center, we redesigned our sepsis order set. We simplified the design, got rid of unnecessary orders, aligned it to clinical workflow, and embedded safety prompts for high-risk populations. And then I analyzed 158 cases, comparing 3 months before to 3 months after the redesign. And the results were encouraging. Sepsis order set use went up over 20%. Step 1 compliance increased nearly 15%, and length of stay decreased and readmissions decreased. This project shared something powerful, that when we improve structure, we improve process. And when we improve process, we create the conditions for better outcomes. So it isn't about asking our clinicians to work harder, but designing the system to work smarter. Because when seconds matter, design matters. Thank you.
Thank you very much, Janelle, for kicking things off. Your presentation is a powerful reminder that in healthcare, timing is not an abstract concept, and really every second can count. Timing can shape decisions, outcomes, the lived experience of patients as well. So it's wonderful to see how your work is taking a high-stakes clinical issue and making it accessible in just 3 minutes for those of us who are not from that field. So congratulations. Are we ready to go on? Or yeah, all right. So moving right along. Our next presenter is Arielle Rousseau, who is earning her PhD in Health and Exercise Science here at Concordia University. The title of Arielle's presentation is Get Strong, One Dish at a Time. The floor is yours, Arielle.
Meet my friend Emily. She's 6 weeks postpartum and has just been cleared by her physician to return to physical activity. Yet she's in pain, not sleeping, the house is a mess, and she's to somehow fit and exercise? How is Emily supposed to recover when she can hardly remember to feed herself? Physical activity is very effective in improving postpartum symptoms like weakness, pain, fatigue. It even decreases the risk of developing postpartum depression, which affects 1 in 4 women. Unfortunately, only 25% of women actually meet the current exercise guidelines for optimal recovery. So wouldn't it be fantastic if Emily could gain strength and improve her functionality with what she's already doing? This is where my research comes in, with blood flow restriction, a method that uses an elastic band around the limbs to partially limit blood flow. When combined with low-intensity exercise, it tricks your body into thinking you're actually exercising at a much higher intensity. Instead of lifting 100 pounds, you get the same benefits by lifting 20 pounds with blood flow restriction bands. That's the equivalent to an 8-month-old child. So this means that Emily could gain muscle strength and endurance just by rocking her child to sleep while wearing these blood flow restriction bands. Now this method is already being used with frail populations, injury rehabilitation, and even with elite athletes, but research has yet to dive into this potentially powerful tool for postpartum recovery. Now Emily's day-to-day involves not only lifting her child, but doing the dishes and lifting up the never-ending piles of laundry off the floor. Do those movements remind you of anything? How about a squat, a deadlift, or a lunge? This summer, I'll be recruiting postpartum women to wear blood flow restriction bands around their thighs while at home with what they're already doing, not adding more to their already full plates. Now, besides household tasks, what's the #1 endurance activity most new parents are doing? Walking, often to get their child to sleep or just needing to be out of the house. I will be hosting right here in Montreal a walking group, bringing new moms together, wearing blood flow restriction bands, and building not only friendships, but muscles like they're training for a marathon. After just 8 weeks, I'll be evaluating the changes of their muscles using functional movement tests such as how many times they can sit to stand in 30 seconds or how far they can walk in 6 minutes. And I'll be complementing that with self-reported questionnaires about their fatigue and pain. Now imagine this. Emily has more energy. She's stronger and her pain is gone. She feels like she can truly take on anything. That is the potential of bringing effective and accessible exercise alternatives to postpartum women like my friend Emily, meeting them right where they already are. Thank you.
Thank you, Arielle. I've seen this presentation 3 times, and every time I get pulled in by the narrative that you build. It's just quite mesmerizing to watch you deliver on what obviously is a passionate subject for you. Likewise, Janelle, before you created a narrative that got us connected with the story. So I think that's a particular useful device to reach, especially an audience that's not from your field. Are we ready to move on to the next one? I'm told that we are. Our next presenter is Ellen van Wijngaarden. Ellen is earning her PhD in mechanical engineering at beautiful Cornell University in Ithaca, upstate New York. The title of Ellen's presentation is Cracking Down on Cracks, a Living Coding for Crack Detection. All yours, Ellen.
I really hate to break it to you, but failure is inevitable. Failure of materials, that is. But how do we know when something like a bridge, an aircraft wing, or even a ship is about to fail? Most of the time, we don't, until it's already a serious problem. Materials slowly develop tiny cracks due to vibration and everyday use. These cracks are often invisible to the human eye, but if they grow unchecked, they can lead to catastrophic failure. Today, I'm going to tell you about a new idea that sounds kind of like science fiction. Materials that sense cracks using living bacteria. Engineers typically rely on sensors, thermal cameras, and inspections to find damage. These methods can be expensive, time-consuming, and don't always catch problems early. My thesis focused on a bold question. What if the material itself could tell us when it starts to break? To explore this, I created a special coating, basically a smart paint. This paint looks ordinary, but hidden inside it are microscopic spores, shown here as the brown embedded speckles in the coating. These spores are a dormant form of bacteria that are not harmful to humans and can survive for years without food or water. When the material is healthy, the spores just sit there doing nothing. But when a crack forms in that material, the coating also cracks. That crack lets in nutrients, which wakes the spores up. They become active bacteria and start producing a visible signal, like green fluorescence or a color change seen here. So instead of needing fancy equipment to detect tiny cracks, the cracks literally reveal themselves. Certain coating recipes prevented that bacteria activation. I tested dozens of recipes to ensure that we have a strong and reliable crack detection signal. You can think of it a bit like this. It's as if the material has tiny living alarm systems built into it. When something starts to break, they turn on and say, hey, look here. Why is this exciting? First, it enables early detection. Small cracks can be found and repaired before they become dangerous. Second, it's passive. This system doesn't need batteries, wiring, or constant monitoring. The bacteria only activate when something actually goes wrong. And third, it's versatile. This kind of coating could be used on structures that are all different shapes, sizes, and materials. But what's especially powerful about this work is that it changes how we think about materials. Traditionally, materials are non-responsive. They just sit there and take damage. My work harnesses nature to move us towards materials that respond and communicate. In the future, we might imagine materials that don't just detect damage, but respond to repair cracks or even prevent damage, helping us build a safer world. Thank you.
Thank you so much, Ellen. I have to say, you got us hooked right away with the title of your presentation. I think it's a great example how a memorable and catchy phrase can make highly technical research immediately relatable and approachable. And there's something quite compelling about the kind of research you're doing that helps us notice problems before they turn into catastrophic failures. So even from the title, this important work sounds like it sits at the intersection of engineering and innovation and public safety. So thank you for sharing that with us. Are we good to go for the next one? All right. Our next presenter is Linda Nung, who is earning her Master of Science in Natural Resource Sciences here in Montreal at McGill University. The title of Linda's presentation is City Rest Stops for Migrating Birds. Whenever you're ready, Linda, the floor is all yours.
This is Mert, a yellow-rumped warbler. This cute little guy could easily fit in the palm of your hand. Every year, he will fly thousands of kilometers between his breeding territory in northern Canada and his winter home in Mexico. As you can imagine, it's an exhausting journey for such a tiny bird to fly for thousands of kilometers. In order to accomplish this, Mert must take breaks throughout his migration journey. During these breaks, he'll be resting in one place for multiple days, eating as much food as he can to replenish his energy before he can fly to his next stop. Now, Mert isn't migrating alone. Every year, he is joined by billions of other birds in their long journeys. But unfortunately, these bird populations have been declining significantly, mostly due to habitat loss. People have replaced a lot of natural forests with cities along their migration routes, and oftentimes, birds end up using city parks as a rest stop. But the habitat in a city park is very different from the habitat in a natural forest. And if birds cannot find enough to eat when they're resting, they won't have the energy to survive the rest of their migration journey. For my thesis project, I want to answer the questions: what specific habitats are birds relying on in city parks? And are these parks providing the right food resources for the birds? Answering these questions will tell us how we can make our city parks better places for birds so that birds that rest here will be able to find everything they need. To do this, we will be tracking 8 different species of migratory birds in an urban Montreal park. We will be using tiny radio tags that the birds will wear, kind of like a mini backpack harness, and these tags will tell us exactly where the birds are going. From this, we can figure out what specific habitats birds use the most and how much space they need to find all of their food. We'll also be collecting poop samples from the birds to see what exactly are they eating. I hypothesize that birds in city parks will have a harder time finding enough food to eat, so they'll have to search for food in a larger area. And I also hypothesize that their diets will contain food items that are less nutritious compared to a bird in a more natural area. This is because cities tend to have very small forests, and within these forests, there are more invasive plants and disturbances from people, cars, and pets that the birds are not used to. Going back to Mert, if he doesn't find enough food to eat when he's resting in Montreal, he's not going to have the energy to survive the rest of his migration journey. Without his help to eat insects and spread plant seeds, our world would look very different. And it would be sad for us too if we no longer heard singing birds anymore. So if we can figure out how to make our city parks better places for birds, we can help Mert and all the other migratory birds on their long journeys. Thank you.
Thank you very much, Linda. Having grown up on a farm about an hour from Montreal where wild geese and ducks would drop by to quench their thirst on our pond, I was particularly curious about this presentation, and it's a good reminder that we share the planet with other species, and not just farmland, but also urban spaces as well. And it reminds us that even urban spaces participate in a larger ecosystem, larger ecological stories. So thank you for reminding us of that. And you know, sometimes every doctoral research is and master's science research is tough, but collecting poop, that takes it to another level, and so kudos and congratulations on that. I was told it was okay to make jokes like that to fill the time, so I hope you know, if you don't like it, you can blame some of my coaches who coached me for this event. Are we good for our next presenter? Yes, we are. All right, so our next presenter is Chaitanya Parekh, who is earning his PhD in biomedical sciences at the University of Massachusetts Chan Medical School. And the title of Chaitanya's presentation is Rewriting Cancer's Code Using mRNA to Unmask Pancreatic Cancer. So take it away whenever you're ready.
Pancreatic cancer is one of the medicine's most deadliest challenges. 5-year survival rate for the disease is just 38%. It is estimated that 7 out of 8 patients would within the next 5 years. Lots of cancer-related deaths in the US by 2030. So take a pause, listen around. The time is clicking, the clock is ticking, and the time is running out. And the time to act is now. Why is pancreatic cancer such a devastating disease? Imagine your computer being infected with a Trojan horse virus. It shields itself so well that your antivirus system cannot detect it. This is precisely what pancreatic cancer does. It hides behind a wall, sends out fake signals so your immune system feels everything is safe. And this is the thing I wanted to tackle during my PhD thesis. I wanted to unmask pancreatic cancer. And for that, I used a molecule known as mRNA or messenger RNA. You might have heard of the term mRNA during peak of COVID-19 vaccines, where mRNA vaccines helped save millions of lives. And we are using the same tool to treat pancreatic cancer. Now imagine if I gave you a USB drive with a set of instructions which will help your antivirus system detect the virus in your computer system. And this is precisely what we are doing with mRNA. With this mRNA, we send precise set of instructions to the tumor. And now the walls are breaking and tumor exposes itself, while immune cells charge towards the tumor while destroying it. And tumor pleads to UMass Chan to stop training more scientists. Does this work? It absolutely does. In mouse models with aggressive pancreatic cancer, when treated with chemotherapy, they unfortunately die within a couple of weeks. With our mRNA therapy, the tumor shrinks and completely disappears. And these mice continue to live up to 300 days and are still alive today at UMass Chan. Are we stopping here? Absolutely not. Me and my mentor, Marcus Rossetti, are going to form a company, Immunoscript, which aims to bring this into first-in-human clinical trials. Our hope is to give a solution to a disease where historically treatments have remained elusive. Thank you very much. Let's defeat cancer. Let's instill hope to these patients. Thank you.
Thank you, Chaitanya, for sharing with us your very important research. I very much like that your title immediately conveys both the urgency of the matter, but also the possibilities that are at hand and within reach, especially with important research like yours. A lot of people will recognize probably by now mRNA types of research, and I think that's very nice to see it placed in a more specific context, and an important one at that. And I think it speaks more broadly to the range of topics that are covered in this 3MT competition, helping society and audiences through a variety of ways, in this case by connecting with complex biomedical advances and research, but using vivid and clear metaphors and making it relevant and clear for those of us who are not from the field. So thank you for that. Good for our next one. All right. Our next presenter is Jessica Tomachik, who's earning her MSc in Earth Sciences at the University of Toronto. The title of Jessica's presentation is a small The title of Jessica's presentation is A Small Crystal with a Big Impact, Unlocking the History of Mars with Garnet. You have the mic, Jessica.
If you were to see this rock on the ground, you'd probably walk right past it. It's small, it's gray, it looks ordinary. But what if I told you that this rock is actually from Mars? Although we have sent rovers, landers, and orbiters to Mars, we have actually never returned a physical rock sample for study on Earth. So how do you study a planet that you've never sampled? The answer? You wait for it to throw rocks at you. When a massive impact strikes the Martian surface, the force is so violent that it blasts pieces of the planet into space, which can eventually fall to Earth as meteorites. Specifically, I want to bring your attention to this Martian meteorite right here that is the focus of my research. It contains the first ever occurrence of the mineral garnet on Mars, pictured in the light gray crystals. But why do we care that garnet has been found on Mars for the first time? Well, besides making beautiful jewelry, garnet is especially useful in geology. Garnets act as time capsules, preserving information related to the changes in temperature, pressure, and chemistry as a rock forms. Commonly on Earth, we see garnets in regions that have been heavily affected by plate tectonics. But this is what is so interesting about garnets being found on Mars for the first time. Our current understanding of the geology of Mars is that there are no plate tectonics, which begs the question, how did these Martian garnets form? As a part of my master's research project, I set out to answer this question, because this is the only sample we have of Martian garnets in the world. I had the amazing opportunity to go to England to use a specialized laser mass spectrometer that works by hitting the sample with the laser to create chemical maps that show me how different elements are distributed and preserved within the garnet crystals. Using these chemical maps and other microscope work, I aim to identify the rock type, learn about the rock's history, and finally compare the Martian garnets to garnets that we see on Earth to try and determine their origin. This work of describing new rocks and minerals on Mars is important because it helps us understand the variety of rocks that we see on Mars. It can provide insights into the evolution of terrestrial planets within our solar system and can help inform future planetary missions. So, what may appear to be an ordinary rock at first glance is in fact a piece of Mars, carrying the record of a planet's history in the palm of our hand. Thank you.
Thank you so much, Jessica. I love this presentation. It's one of those wonderful topics that takes something, really tiny and turns it into a doorway to something much bigger, vast, beyond our everyday, our immediate. A small crystal, a whole planet, and a scientific research that bridges the two. It's exactly the kind of scale-shifting research that makes this 3-minute thesis competition so exciting. And I just love when Jessica was telling her about going to England to use all this technology. You could see her eyes light up. You could see how passionate she is about her research. So thank you, Jessica, for sharing that passion with us. We're good? All right. Our next presenter is Hannah Wiswell, who's earning her PhD in mechanical and aerospace engineering at Princeton University Graduate School. Hannah's presentation is titled Avian-Inspired Engineering, Learning from Birds to Design Safer Aircrafts for Human. Go ahead, Hannah.
For more than 500 years, since before Leonardo da Vinci first sketched the invisible airflow over a bird wing, we have turned our gaze skyward, looking to birds for answers to how to build aircraft and fly with the masters of the skies. Even after all that time, though, we have more questions than we do answers, especially as our requirements for aircraft become more challenging to meet. For example, how can our aircraft safely and efficiently carry an increasing number of passengers and cargo thousands of miles across the sky and remain aloft and stable in increasingly unpredictable wind environments? If we again look to the sky, we see that many birds can easily soar across vast distances, carry prey heavier than their body weight, and maneuver through gusty winds without faltering. So how do they do it? And how can we do it too? Well, Da Vinci also wrote that to understand bird flight, you must first understand the anatomy of the bird and its wings. If we look at one such bird, like an eagle, for example, one group of feathers on its wings stands out in how they interact with the air around them. These feathers, the wingtip feathers, unlike any other feathers on the wing, simultaneously separate, bend, and twist during flight. Their unique material properties and simultaneous motions are so complex that no one, even after 500 years, has been able to determine exactly how these feathers affect the airflow over a wing, enabling birds like eagles to fly how we want our aircraft to. But what if we break down that complexity and study these motions individually first, and then together without losing any of the important physics? I do this in my PhD by using bio-inspired design at the intersection of engineering and biology. I create engineered wing models like you see on the slide, which preserve aspects of the biology like wing shape. And I use multi-material 3D printing to create engineered wingtip feathers to capture each individual parameter: separation, bending, and twisting. I then place these different models in a wind tunnel and use a combination of cameras, particles, and even lasers to visualize and measure the airflow over each wing. So far, I found that wingtip feathers significantly impact the airflow over a wing. But what's more, separation, bending, and twisting each have their own unique impacts, emphasizing the importance of studying each of these parameters separately. For example, I found that alone, feather separation can control a wing's ability to remain aloft. But when you add bending and twisting, you also increase its maneuverability. So together, these motions make a wing robust to and able to take advantage of sudden and unpredictable changes in the air. By using bio-inspired design to create engineered wingtip feathers that not only preserve aspects of the biology, but are also applicable on engineered wings, I gain insights into bird flight and can design safer aircraft for humans to fly in any condition alongside the masters of the skies.
Thank you very much, Anna. There's something riveting and appealing about research that learns from nature, not just to replicate it or to admire its beauty, but to solve real human problems. And your research reminds us that how often innovation begins with an acute sense of observation, in this case by watching birds and asking what they might teach us about flight design and flight safety. So quite compelling, and thank you for sharing that with us. Well, we've made good time. We've kept good pace, so we're a little bit ahead of our schedule, but we'll take, I believe, a well-deserved break for participants and audience members as well. We'll reconvene at 2:55, so we have just a little under 15 minutes, and we'll see you back at 2:55 for our next presentation. So thank you, and we hope to see you back after the break. Okay, I'll do that. Welcome back, everyone. I hope you had a nice short break. We were fighting for the last Timbits here, so hopefully you didn't have to do that at your end, wherever you're zooming in from. And I hope most of all that you're ready to hear more amazing research from our presenters. Our next presenter is Esther Apula, who's earning her PhD in molecular and cellular biology at Stony Brook University. The title of Esther's presentation is RSVP Required, Infections Denied Entry. Good luck, Esther.
We can all agree that getting sick is the worst. The fever that drains you, the chills, and that cough that makes every breath hurt. Now I want you to imagine all of those symptoms unfolding because you breathe in a few bacteria. Not thousands, not hundreds, just a few. That is tularemia, a disease caused by a bacteria called Francisella tularensis. In nature, one can get tularemia from tick bites, contact with infected animals, or you can breathe it in if it's dispersed in the air. When inhaled, it causes a potentially fatal pneumonia. Now, because a tiny amount of the bacteria can cause disease and how easily it can be dispersed in the air, Francisella has drawn military interest in the past. During World War II, several countries investigated tularemia as a potential biological weapon. Until today, it remains classified as a Category A biothreat. Now, despite decades of research, there is currently no approved vaccine against tularemia. And that's where my research comes in. I'm working with the weakened Francisella strain, where a protein linked to its virulence has been removed, so it doesn't cause severe disease like the normal strain would, but activates the immune system. Our immune system is made up of cells that are supposed to detect and eliminate threats like bacteria. Some of these cells act like security guards. They patrol for threats and alarm the body. And some act like garbage disposals. They clean up infections. But Francisella flips the script. Rather than get destroyed by these cells, it uses them as safe houses to multiply and spread. So by the time these cells ring the alarm, it's already too late. Studying this weakened Francisella strain in mice, I'm uncovering how the bacteria outsmarts the immune system and pinpointing key immune cells it exploits. With this, I'm able to train the immune system to better target Francisella. With this strain as a potential vaccine candidate, the goal is to prepare the body in advance to quickly recognize Francisella and deploy the right immune cells to clear the infection. Because we all deserve to feel protected against natural infections and potential biological threats. And we know the best defense isn't panic, it's preparation. Thank you.
Thank you so much, Esther. I don't know about you, but I've never been as scared of a single bacteria all of a sudden. And I have to say, Esther, your opening narrative was so relatable. I think we could all remember a time where we've been sick and sniffles and all of that, but at the same time, your title is very playful, but also very evocative. It triggered all sorts of mental associations and connection. It suggests some gatekeeping mechanism, point of entry, some possible mechanism for prevention, and that in just a few words. So really well-chosen title that can connect rapidly to a broad audience. So well chosen and great delivery. So thank you for sharing that with us. We're on track for our next presenter. All right. So our next presenter is Abouzar Qassemi, who is earning his PhD in food microbiology at Université Laval. The title of Abouzar's presentation is Microorganisms as Tools to Unlock the Hidden Value of Fruit Byproducts. Whenever you're ready, Abouzar, it's all yours. Abouzar, I think you're muted. You need to unmute yourself. Do we know what's going on? Yep.
Hello, everyone. Imagine if we could turn trash into treasure just by letting some tiny creatures called microorganisms do their thing. They are tiny, they work 24/7, and they never ask for a salary. Dream employees. Every year, as fruits are processed for juice, jam, and sauce production, a large amount of byproducts is generated. Instead of letting this waste damage our environment, our project aims to convert it into nutrient-rich supplements. As a matter of fact, our goal is to take what was considered waste and give it a new life that benefits both our planet and our communities. So it's not trash, it's just misunderstood. So how do we make this transformation happen? We begin with a step-by-step screening of microbes naturally present in these materials to identify the most promising ones for a natural transformative process called fermentation, like yogurt production. In fact, screening is basically like a job interview for microbes, where we hire the right candidates to work as microscopic chefs. Then we work on finding the best conditions for the activity of the microorganisms to produce a well-balanced nutrient-rich supplement. In the final stage, we simulate the digestive system in the lab to see how these supplements interact with gut bacterial community. It's digestion, but in glassware. Same concept, fewer unpleasant surprises. This will help us understand if our innovative supplement can potentially support the health of the host. So what can we expect from this project? In terms of health, this transformative process can improve the nutritional value of fruit byproducts through the action of microorganisms that produce and release a broad range of beneficial natural compounds. It also enhances their digestibility by breaking down the tough plant structures. On an environmental level, turning fruit byproducts into useful supplements means less waste in the landfills, helping reduce emissions that lead to global warming. Economically, this transformation opens up new revenue streams and creates job opportunities. It's like giving waste a LinkedIn profile. Overall, our project is about finding a practical solution to reduce food waste, because the only thing that should be wasted is time, not food. Thank you, and if you ever feel useless, remember, even fruit peels can get promoted. Thank you.
Thank you, Abouzar. I have to admit, I was particularly looking forward to this presentation, having spent the last 2 decades at the intersection of sustainability and food marketing. Food waste is definitely a big concern, and whenever I talk to students about the outlook and the impact of our food industry, it's easy to come away with a gloomy sort of outlook on things, that things are really bad. But what I love about your research, Abouzar, is it's pointing the way towards very promising applications that can help us address wicked issues, wicked problems like food waste, so that the outlook is not as gloomy as we might think. Moreover, I think your research really encourages us to rethink our definition of food waste and what exactly is food waste. And it's a great example of work that connects different fields from microbiology to sustainability and innovation. So very much appreciated your presentation. Also, a little note from a communication perspective, the ending of Abouzar's presentation was also remarkable in terms of, again, coming full circle and connecting all the dots, you know, alluding to fruit peels and whatnot. Are we ready for our next one? Yes, we are. Okay, so our next presenter is Ashley Baston, who's earning her PhD in biological sciences at the George Washington University. The title of Ashley's presentation is Trained to Destroy, Uncovering the Causes of HIV-Associated Dementias. Ashley, it's all yours.
What does the term memory mean to you? If you ask me as a graduate student, I would say that memory is the thing that magically disappears right before all of my exams. But if you ask me as someone who studies the immune system, I would say that memory is a complicated way in which your immune cells work to protect you, which sometimes can go very, very wrong. For people living with HIV, the term memory takes on a more complicated meaning. Up to 50% of people living with HIV experience HIV-associated dementia, a serious condition that can result in memory loss. Currently, treatment options are extremely limited, as the exact cause remains unknown. However, my research aims to find that cause, exploring a different kind of memory, that of the immune system, as a potential path to a cure. Trained immunity is a newly discovered form of immune memory unique to anything else in your body. Think of this process like running a sports team. You have a coach, you have your team, and you have an opposing team. In trained immunity, that coach is a protein called a training stimulus that interacts with your immune cells, your team, to respond against potential threats, the opposing team. Usually, this system works very well to keep you healthy. However, during HIV infection, trained immunity can go haywire, launching a massive inflammatory response that damages the connections between those cells that facilitate memory. Kind of like a bad coach overtraining an athlete to the point of injury. My research investigates the HIV protein NEF as one of those bad coaches, a harmful training stimulus that interacts with the critical support cells in your brain to sever the connections between the cells that facilitate memory. To test this, we expose both mouse and human brain cells to NEF, and we look at metrics like cell size and shape and how they respond to stress. We use techniques like fluorescence microscopy, labeling each of these cell types with a different color, allowing us to see exactly how NEF is facilitating these harmful changes. Our results are clear. NEF is a harmful training stimulus that induces inflammation and changes the way that these cells respond to stress. In my work, the term memory retains a double meaning, serving as both the trigger and the target. But it's my hope that through this work, perhaps memory can even be part of the cure. I hope that this research inspires cures and treatment options for HIV-associated dementia, but I also believe that it can go even further, inspiring potential treatment options for similar conditions like Alzheimer's disease, bettering the quality of life for millions of people around the world, all through memory. Thank you.
Thank you so much, Ashley. I love how Ashley immediately connected with all of us by referring to her memory giving way around exam time. We can all relate to that. And that's a great example of a universal. When you're able to tap into something like that, you immediately are able to connect with every member of the audience because it is a universal experience. And Ashley's title really suggests both intention and consequence. I didn't expect to learn so much about different kinds of memories and the immune system. Definitely dementia, Alzheimer, cognitive impairment, degenerative cognitive diseases are on the rise. So very timely and appropriate research. And thank you for sharing that with us, Ashley. We're good for our next presenter. Thank you. Our next presenter is Mariam Afkari, who's earning a PhD in chemistry at Université de Sherbrooke. The title of Mariam's presentation is Understanding the Effect of Oxygen-Nitrogen in Titanium Oxynitride Supercapacitor. Bonne chance, Mariam.
Well, I think most of you enjoy watching Olympic Games or sports, and you probably know about Usain Bolt. He is an Olympic champion for 8 times. He's a sprinter. He runs 100 meters in less than 10 seconds. At one time, people joked everyone that runner that fast might turn to dust. But don't worry, actually he's alive and he's just doing fine. What makes him so special is he releases huge charges instantly. He's like an electric car. Imagine you can charge it for 30 seconds. This is fantastic. But you can drive it only for 5 kilometers. Now, on the completely opposite side, there is marathon runner legend, Eliud Kipchoge. Eliud can run over 40 kilometers in almost 2 hours without stopping. Actually, he has less charges, but he releases very slowly for longer times. Imagine the same electric car again, but this time you need to charge it for longer times, like 6 hours. But at least you can drive it for 40 kilometers. So this is our energy rule. Batteries are like Eliud Kipchoge. They charge slowly, but they last longer. Capacitors are like Usain Bolt. They charge so fast, but run out so quickly. Now humanity keeps asking this question, can we have both at the same time? Charging fast and using for the longer times? Well, I'm a chemist. My job is basically cooking. So in my PhD, I started with a stable compound, and that was metal oxide. It wasn't chargeable. So I tried to add a new element on it, and that was nitrogen. And I cooked them at high temperature, and I changed them in atoms levels. The material that I created can charge fast and last longer, and that is why we call it super capacitor. This is very important because those materials are going to use in electric cars, which run on by electricity, not gasoline. Recently, a lot of countries like Canada moving forward to electric cars because they are environmental friendly. Actually, the material that I created not only charge fast and last longer, but also it is environmental friendly. Therefore, in the future, your electric car won't need to choose between a sprinter or marathon runner. Probably it will have both. And this feature might start from our lab with my cooked atoms. Thank you so much.
Thank you. Merci beaucoup, Mariam. I have to say, in my own wedding vows, I managed to sneak in Dumbledore and Shakespeare, but you've really bested me, Mariam, by linking Usain Bolt to advanced materials chemistry. So that's quite a feat. And all seriousness, one of the pleasures of this competition is hearing about highly specialized research that in your daily life you might have overlooked, you might not have read the press article or listened to the news piece because it's something you couldn't relate to or that's not in your sphere of experiences, but you made this highly relatable, Mariam. Thank you so much for that. We could easily hear the precision of your knowledge, the technical depth that you bring to your work, and how you made it all highly relatable and accessible. So thank you very much for that, and congratulations. We're on to the next presenter. So our next presenter is Nia Ismailzada, who is earning her PhD in biological sciences at Fordham University. The title of Nia's presentation is Motivation in the Brain, the role of dopamine D5 receptors in motivation-related behavior. Nia, whenever you're ready, the floor is all yours.
We all know how it feels to have absolutely zero motivation to do things that we know we have to do. Some people pursue goal after goal. Others don't even have the drive to get started. Not because they're lazy, but because something in their brain's motivation system has gone wrong. Depression, ADHD, schizophrenia, and many more neurological disorders share something in common: changes in motivation. Motivation controls everything that we do, starting from the simplest accomplishments, ending with biggest dreams. But what controls motivation? The answer lies inside of our brains. Right now, as I speak and as you listen, millions of neurons are constantly communicating with each other, transforming your thoughts into action. One of the brain's main messengers in this process is dopamine. Dopamine acts like a signal. It's a neurotransmitter that tells your brain what is worth pursuing and what is not. But dopamine cannot work alone. It needs special proteins in the brain called receptors. Receptors are sensors. They detect dopamine and translate its message into behavior. When these receptors go wrong, so does motivation, contributing to the same neurological disorders mentioned before. I studied the least understood dopamine receptor D5. To understand dopamine D5 receptors function in motivation, we generated a special mouse model genetic mouse model in which we deleted D5 dopamine receptors from the areas of the brain involved in motivation. We then tested these animals in different behavioral tasks to understand how the motivation changed. In one of the tasks, mice have to perform progressively harder to reach a reward. With each step, the task becomes more difficult. And guess what? At some point, animals give up. But the point when they give up can tell us how motivated they are or how much effort they are ready to put into the task. Surprisingly, deleting D5 dopamine receptor from the area of the brain involved in motivation did not reduce motivation. It increased it. Animals became more motivated and more willing to put effort into the task. This may suggest that dopamine D5 receptor may act as a brake, holding the drive in check. Understanding how dopamine receptors regulate motivation will open doors in neuroscience into understanding how the brain regulates motivation. But most importantly, remember last time you had lack of motivation? That is a fleeting feeling for most of us, but it is a permanent reality for millions of people struggling with neurological disorders. Understanding how the brain regulates motivation may one day change the reality for them by giving them back something that we all need, their motivation. Thank you.
Thank you so much, Nia. I have to say, every now and then you read things in the press or in a news report about dopamine, and it's often reduced to a soundbite or a catchy phrase, and you've managed to unpack that way beyond the catchphrases and the headlines. So thank you for demystifying a lot of that. Something caught my attention during Nia's speech and how she snuck in there that she's studying the least understood receptor. And I love how she managed to underscore that. There's nothing wrong about tooting our own horn, so to speak, and say, I'm actually studying something that not that many people have studied before. I think it's a subtle but effective way to underscore the importance of what we're doing. And especially at the end, Nia, when you tapped into our sense of empathy, by pointing out that millions of people suffer from permanent state of lack of motivation. I think those of us who have struggled occasionally with motivation, to imagine that some people live in a perpetual state of lacking motivation, we can all relate to that and feel empathy for these people. Are we good to go with our next one? Yes, we are. So our next presenter is Christina Stallings, who's earning a PhD in immunology at the University of Pennsylvania. And the title of Christina's presentation is A Piece More Beautiful Than Art. Good luck, Christina.
One day in high school AP biology, I was learning about the immune system, and my instructor showed me this chart. When you're sick with a germ, your immune response activates and fights disease, giving us the first peak we see here. And if you're exposed to the same germ, your immune response will reactivate, only this time it's faster, stronger, and more efficient, to where you don't even realize symptoms, giving us the second peak we see here. And this is the basis of your immune system, designed to protect you from disease for all of your life. Yet, I thought, this chart can apply for all diseases. So I asked, well, what about HIV? And my instructor responded, excellent question. With HIV, this chart is erased. What makes the virus so dangerous is that it attacks the commanders of your immune system, the helper T cells. Upon infection, HIV inserts its DNA within the cells and reprograms them to create more virus clones. These clones then spread and repeat the cycle, killing more and more helper T cells in the process and weakening your immune system. Scientists created something called antiretroviral therapy, better known as ART, in response to this. And it's an excellent drug that forces HIV to stay asleep within the cells, changing HIV from a death sentence into a manageable condition through the daily pill or the monthly injection. While this is good progress, I have to ask you all, why should we stop here? ART still has its limitations. If drug intake is interrupted, HIV will reawaken and continue from where it left off. So what I propose is a therapy that can turn your own cells to be the living drug that beats HIV. And this is what I do. I take the soldiers of the immune system, the killer T cells, and I give them an HIV protein detector that guides them to the HIV protein expressed on the infected cells and helps them do their job of putting HIV to sleep. So in my lab, I create these super soldiers, and I put them in a dish alongside the infected HIV cells and let them sit to test to see if they can perform their job. After a few days, I get my dishes and I put them in the machine to determine the level of HIV protein expression. And in my experiments, I do see lower levels of expression, meaning that my super soldiers were successful in doing their job, returning us back to our chart. 40 million people are living with HIV, and with my research, we can give them a one-time treatment to keep HIV asleep within their bodies forever. No more pills and no more injections. And with that, I hope we can take away that we are still pushing towards progress. We did it once to make HIV manageable, and we'll do it again to give it a proper cure. Thank you.
Thank you so much, Christina, for sharing your very vital research in so many ways with us. So many great communication tools that you use. First of all, the title, I think, is what a great title should do. Create in the audience a sense of wanting to know more, wanting to hear more, so getting us hooked, grabbing our attention right away. But what I particularly like is you held our attention. You held our attention with metaphors, the soldier in our immune system by referring to the T cells. So right away, what is a complex phenomenon that you understand because you're knee-deep and you understand this research, you made it accessible through those metaphors for the rest of us. And again, the importance of your research, and this is what is so mind-blowing. I told you you'd be uplifted by listening to all these presentations. 40 million people here can benefit from this research. And I think this is so amazing what our graduate students are doing, not just for the 3MT competition, but across Northeastern regions and all over the world, is working on important research that can have true positive impact for the whole of society. So well done, Christina. All right. I'm just waiting for the signal that we're good to go. All right, I was ready to do a little song and dance and tap dancing number, but I guess I won't. I'll spare you that difficult experience. Our next presenter is Zahava Hirsch, who's earning her PhD in biomedical engineering at the Stevens Institute of Technology. The title of Zahava's presentation is Improving Turning Balance with Music Biofeedback. Take it away, Zahava.
Imagine this. Your grandfather is heading out and he realizes he forgot his keys. He quickly pivots to go back inside, but that sharp turn catches him off guard and he falls to the ground. This is not just a hypothetical. 1 in 3 older adults falls every year, and a fall during a turn is 8 times more likely to result in hip fracture. What's worse, nearly 25% of older adults who suffer a hip fracture will not survive beyond a year. Now, despite turns being a trigger for fall injury, there is very little known about the biomechanics of turning and how to improve turning stability. That's where my research comes in. In studying turns in over 20 older adults, we have found that turns do result in riskier balance states, and that older adults tend to use person-specific balance strategies when navigating turns. This emphasizes the need to develop personalized interventions that can train turns in a safe and engaging way. To that end, I have been working on improving turning stability using an innovative approach, music biofeedback. Music biofeedback conveys real-time changes in balance metrics using precise changes in musical sound. This, 1, leverages the health benefits of musical interaction. 2, does not distract other senses required for safe turning, such as vision. 3, provides real-time corrective information. And 4, music biofeedback can be personalized per individual depending on their unique needs. For example, if an older adult needs to work on widening their step width, a metric that is often challenged during turns, we map changes in their step width to changes in musical pitch. This means that while they're walking and turning, if their step width gets too narrow, the pitch of the music that they're listening to increases, informing them that they need to widen their steps. Similarly, if an older adult needs to work on posture control, another metric that is associated with falls, we map changes in their posture angle to changes in musical pitch. So while their posture is upright, they hear a pleasant musical sound. However, as they begin to lean forward and that posture angle increases, the pitch of the music that they're listening to increases, informing them that they need to self-correct. To date, 15 older adults have trained using our music biofeedback system, and all have demonstrated immediate improvements. 10 of these older adults have also completed a 4-week-long pilot clinical trial, resulting in retained improvements post-training. The purpose of this research is to empower older adults to move with confidence. By improving our understanding of the biomechanics of older adult turns, we can develop personalized interventions to help ensure that a simple turn does not result in tragedy. Thank you.
Thank you so much, Sahava. This is amazing research. I'll fess up that I've benefited from biofeedback training after a cardiac event, so I know how important that whole approach is, but what incredible advancement and innovation to use music to this for the service, or to put that to the service of aging adults. And love how you open your presentation with a fall during a turn is 8 times more likely to cause hip fracture. I mean, that's grabbing, that's vivid, that's tangible. We can wrap our minds around that. This is also a beautiful example of interdisciplinary research using all sorts of approaches. That's just quite innovative. We can hear in your passion and your description, movement, sound, health, innovation, engineering, all coming together to help address real problems. So congratulations. And on a personal note, as someone who appreciates big, bold glasses, I love yours. I think they're becoming, and you wear them very, very well. All right. Are we good to move to the next presenter? All right. Our next presenter, our final presenter, I should say, today is Abigail Fry, who's earning her PhD in Biological Sciences and Public Health at Harvard University Graduate School of Arts and Sciences. The title of Abigail's presentation is Uncovering the Unknown, Identifying Drug-Resistant Tuberculosis. Good luck, Abigail.
Imagine this. 1/4 of all humans have a deadly pathogen inside them, waiting to strike. Every year, 10 million people will fall ill. 1 million of those people will die. This is not the plot of a science fiction novel. This is the reality of the deadliest infectious disease in the world, tuberculosis. We don't hear much about tuberculosis, or TB, here in North America, but essentially it is a bacterial infection of the lungs that spreads person to person through the air. And it can be treated. However, 1 in 25 cases of TB is drug resistant, meaning that the standard treatment regimen will not work. The good news is, many forms of drug-resistant TB can be treated with alternative antibiotics if they're properly diagnosed. The most efficient way to perform this diagnosis is by screening the DNA of the bacteria for drug resistance mutations. But the bad news is, we don't know all of the mutations to look for. In my research, I am working to identify those unknown mutations. To do this, I use a mix of computational and experimental techniques. In the computational work, I analyze big data sets of DNA sequences from TB bacteria from patients around the world. And with this approach, I identified a new gene that, when mutated, is associated with an increased rate of treatment failure. But the only way to know for sure whether these mutations cause drug resistance is in the lab. Here, I work with the actual pathogen, genetically engineering it to have the mutations I identified in the patient samples. Then, I test the mutated bacteria's ability to grow in the presence of antibiotics. And I found that these mutations do cause drug resistance. In fact, the bacteria with the mutations require twice as much antibiotic to be killed. Altogether, this work illustrates the importance of combining computational and experimental techniques, as these findings can now be applied to improving drug resistance diagnostics. So now imagine a world in which no one dies from drug-resistant tuberculosis. I hope that by identifying the mutations we've been missing, we are getting a little bit closer to making that our new reality. Thank you.
Bravo, Abigail, for bringing our competition to a close, for now at least, while our judges deliberate with such a powerful and wonderful presentation. You brought forth definitely one of the central purposes of research, which is to make the unseen visible, to make the unknown knowable, to make the urgent and the important understandable. And definitely drug resistance is both a scientific and a very important public health challenge. It even touches my field, which is food. And I very much appreciated how you started with some pretty dire statistics. 1 out of 4 people will have a deadly pathogen, 1 million will die. But before it was too gloom and doom, you pointed out that there is good news through proper diagnosis and use of alternative antibiotics. So it's nice how you flipped that around before we got all too depressed or feeling too gloom and doom. So congratulations and good luck on your very, very important research. So now I understand we will take a break. After that, when we come back, it'll be a very short break just so we can get matters all aligned and confer with our judges that everything is on track. And when we come back, we'll open the voting for the People's Choice Award. So if you want to vote for your favorite presentation, make sure to stick around and be here at 3:35. So very short break, 5 minutes, and we'll see you back here very shortly. Thank you. All right. Welcome back, everyone. I hope you're still surfing on that high, that uplifting feeling from listening to all these amazing presentations, all the vital and critical research that we heard about, and that surprisingly we understood, because our presenters were just so amazing at breaking it down in a relatable and understandable way. So before we go any further, a big round of congratulations to all our finalists. Yes, please pat yourselves on the back. Take a moment and savor the moment. You've done it, and good job presenting complex research in a clear, compelling way is quite difficult. To do so with confidence is even more challenging, so congratulations. At this time, voting for the People's Choice Award is now open, I believe. Let me just double check. Yes, it is. And our judges will begin their deliberations. Audience members may vote now using the link in the chat box, or by using the QR code on the screen that you see here. Please note that you may submit only one vote. No matter how badly you like more than one presentation, you are only allowed one vote, and you're allowed to vote for a single favorite presentation. So voting will remain open until 3:45. So you have just about a little under 10 minutes to get that voting done. We will now move into a brief Q&A with our presenters. So if you have a question for a specific presenter or a broader question to the overall group, please enter it in the chat, or feel free to unmute yourself and voice your question. If you are here in the live audience, simply raise your hand and we'll get you a mic to ask your question. This is always a fun part of the event. Our presenters' stress level is going down. Wonder what that's doing to their dopamine receptors and their breathing and their immune system. So now they can step back, enjoy, reflect, and address your questions. So again, if you have a question, you can enter it in the chat box, or unmute yourself and ask it. And if there's no question, I'll go with some of mine. Whoever of our presenter wishes to answer that question. A lot of PhD and master's students, when they get started, they're unsure about the process of choosing a topic. Sometimes you work with an advisor's already working on a project, and that's what you insert yourself into. But I'm curious, for some of the presenters who are listening, how did you go about landing on such amazing topic? I wonder if anybody wants to chime in and just speak a little bit about the process of picking your topic, refining it, how that was, whatever help, inspiration you might have gotten along the way. And if you want to express gratitude to someone who might have helped you get there, feel free to do so. This is a great forum to do that. I think we have a hand up, do we? Yes, Christina. Christina? Yes, Christina, you want to chime in? Please do so.
Absolutely. Can you all hear me okay?
Yes, we can. And thank you again for your wonderful presentation.
Oh, absolutely. So regarding your first question about deciding on what to commit to, because it is a big commitment. The PhD is not short. You are dedicating a good portion of your life to this piece of paper. So for me, as I mentioned in my talk, actually, it is true. In my high school AP biology class, I was introduced to the immune system and I fell in love. But even more profound was that there are certain diseases that seem primed just to overcome your intricate system. And I thought, in this day and age, with our technology, we should be able to go further. Why should we stop where we currently are now? And that's one of the main goals of science, is the pursuit of knowledge. But not only that, to use your creativity to pursue even greater things beyond what you started with. And so coming here to Penn, it was such a blessing to find innovative minds, creative minds, to introduce me to the science and innovative ways to create new innovations to improve medicine. And I just wanted to start with the disease that brought it all, which was HIV.
Wow, thank you. And I have to admit, we could feel your passion, and it's nice. It's interesting to see that your interest was sparked a while back, right? And you've built up to this, and you've accumulated all this fantastic wealth of knowledge that obviously seems to all be coming together in this moment or in this leg of your journey. So congrats on that. Anybody else has thought on the choice of a topic or what led you there? I see Ariel in the audience has raised her hand. You want to come up to the mic, Ariel?
You have Hi. So my kind of motivation to pick my topic was that I'm a mom of two, and I'm also an athletic therapist, and I specialize working with actually prenatal and postpartum women. And I just find when they come into clinic, kind of week after week, there's a lot of that mom guilt of I didn't get to do my exercises, I'm feeling strong, I'm tired, I don't know what to do. So my motivation was really to find a topic that could kind of help them in that in-between stage of trying to find time or not needing to look beyond time to get strong and to feel good about themselves. So yeah, that's kind of where it came from. Thank you.
Thank you, Ariel. And I have to say, I'll fess up because I've been participating in these presentations. I've seen Ariel's presentation, and every time your smooth delivery, and now that you explain it, I've never heard you explain it this way, but now I think I can understand where that smooth and effortless delivery comes from, because it is deeply personal. You are a mom and you've experienced some of these things, so now I get a little glimpse on that process. And I know it doesn't come easy. You've practiced, you've rehearsed, but you make it look also effortless, as many of our presenters have as well. I see more hands that have gone up. I can't quite see the screen, so I don't know in which order. I'll let the...
Ashley, I believe Ashley Baston, you had your hand up first.
All right.
Thank you. I actually think my introduction to immunology and virology was almost the complete opposite of Christina's, where the first time I was introduced to immunology, I looked at it and I said, oh my gosh, this is really hard. I don't ever want to do that again. And then when I started my PhD, I was very enticed by working with viruses. I've always had, since childhood, an appreciation for finding out what causes disease. So finding out I was going to work with HIV, fantastic. Finding out that that would require a ton of immunology, not so fantastic. But over time, as I became more involved and I learned so much more and I got to do experiments of my own, I began to gain such an appreciation for not only how disease works, but all of the things that need to go exactly a certain way for that to happen and how much that relies on the immune system. And now getting to do things like this, I can take this newfound love and appreciation of immunology and I can make it understandable for other people so that hopefully when they encounter immunology with or without the kind of virus context, it's not the same reaction I first had, which is, wow, this is so challenging, but instead it's, this is so cool. I hope I can learn so much more about this. And of course, I absolutely love and appreciate my current research, and I found such an appreciation. And I hope sharing here today, I hope other people kind of have too.
Thank you for your transparency in admitting that, it was not love at first sight, perhaps, with immunology. But, what they say, if it were easy, we probably wouldn't want to do these things. So whatever is worth doing is often hard. And I think it's fantastic that your feelings turned around and you persevered. And thank goodness you did. I see other hands up, but I don't know in which order they came up. So
Jessica Tomacic. Jessica, we'll hand things off to you.
Great. I just wanted to congratulate everyone. Everyone did amazing, really fantastic presentations. But how I got to start my research, I've always just loved science and being curious, and I love just nature and how the world works. And I found it really fascinating when studying earth science that from such a microscopic crystal, you can learn about the geology of, let's say, a whole planet. And I think what's really important in general too is just science communication, because I firmly believe, what's the point of science if not everyone can understand? So that kind of led me to the University of Toronto and working with my great supervisor, Dr. Kim Tate, where I do my research with the Royal Ontario Museum in Toronto. So I constantly get these amazing opportunities to share my passion for the earth sciences with all sorts of audiences and inspiring others. So yeah, thank you.
And you did inspire us as well, so thank you for that.
Next up, we have Janelle Harding.
Yes, thank you. Choosing sepsis as a topic was quite easy, actually. At my healthcare organization that I work at, sepsis is the second leading cause of mortality where I work, as well as the leading cause of readmissions. So it's definitely a hot topic to try and target to make improvements. Additionally, we were in a period of time where our order set review was due. So looking at the order set, making sure things were still aligned was again an optimal chance to do that. In addition, culminating at the same time, the Centers for Medicare and Medicaid Services incorporated that SEP1 core measure into an additional program that they have that now tied financial reimbursement to how our compliance was. So again, really targeted sepsis as something that we needed to improve, and so that's why sepsis was quite easy as a choice.
Wow, wow, and congratulations for the impact you're already having with the things that you're doing. It's just quite impressive. Who's next? I think we have time for a couple more comments.
Next up, we have Sahava Hirsch. Sahava?
Hi, I wanted to thank you. I really appreciate that question. My journey to my PhD was not linear. In my undergrad, I did biological studies and religious studies, and I minored in LGBTQ studies and studio art, so I was really all over the place, and I did not know what I wanted to do. And I think what I like to kind of focus on is that it's okay to not know what you want to do. I kind of continue to just follow my passions. I took a break from the sciences for a while. I worked on a documentary. I did some art stuff, and I ended up getting my master's degree in biomedical engineering as part of a late entry program because I didn't have an engineering background. And I persevered, and I was really fortunate to meet my advisor, Dr. Antonia Zafriou, who shares a lot of the same values as me. And we were just like able to combine arts and sciences in a really beautiful way, and in a way that I really appreciate, and really combines so many of my interests, where we put we put the users first. We do very user-centered design to make sure that what we're creating is for a population that will actually use it and appreciate it, and we're not just doing science for our sake or for the sake of publishing. So I think my takeaway is that if you stick to things that bring you joy, if you stick to things that kind of instill passion and make you excited, even if that journey is not linear, in my experience, it does ultimately pay off. So you should just kind of keep searching for what brings you fulfillment.
Oh, thank you for this. I wish I could just package all this and share it with all graduate students. As an academic and an administrator, there is beauty about a very linear approach, somebody who's focused and gets their journey done, and I applaud that as well. I had a meandrous background, and I think you underlined for us how valuable, doing many things and then eventually focusing, that can enrich your journey in other ways as well. So thank you for sharing that. It's fantastic to see, really.
Next up, we have Hannah Wiswell.
Yeah, I definitely agree with Sahava that it oftentimes happens that you don't know what you're going to do. And I think the same is said for me. I started off as a music performance major and switched to chemical and then went into mechanical. And the one constant throughout the journey was always David Attenborough. Whenever I was stressed, and particularly around exam season and even high school, I would buy like a mega pack of raisins and eat raisins and watch Blue Planet or Planet Earth while I was studying. And I didn't even know that bio-inspired design existed until I kind of stumbled upon a butterfly effect person, which was my undergrad aerodynamics professor. And he explained it to me, and I had just no idea that it was real. And then I just began kind of noticing the birds that were the seabirds by my home and the birds that were flitting around the Kate May Lewis ferry, and I was really fortunate to find my advisor and just was like, I love birds. How do I also do engineering? And since then, since meeting her and all these other butterfly effect people, there's not been a day that goes by that I'm not excited by what I'm doing. So you'll find your way.
Thank you for sharing that. I wish we could package all these stories into a book. I think future graduate students would really enjoy this. But your presentation, as I was listening to you and your study of birds, I was reminded, growing up on the farm, we had goats, and I was fascinated watching the goats eat just about anything, flowers, grass, hay, and I would try to approach the goats and watch them eat. And I was fascinated. What happens? Like, how come they can eat all this stuff? And how does it taste like? And years later, I started studying cocoa, and I traveled the world tasting different cocos and visiting different plantations. And in one particular instance, I tasted a particular chocolate called criollo from Venezuela. It's a type of chocolate or cocoa bean that's called porcelana because the bean stays fairly white as it's roasted. When I ate that chocolate, I became the 5-year-old boy watching the goats, because in that moment I said, this is what flowers and grass taste like. That chocolate had so much floral complexity, and it took me back. And the point that I'm belaboring to make, obviously, is how much there is to learn about observing nature, about trying to relate, about trying to apply what nature has to teach us. So thank you for this. And we have time for one last comment before we turn it over, I believe, to the judges and announce the winners.
Next up, we have Ellen Van Weingarten.
First off, a huge thank you to the organizers for putting on this incredible event. What I found particularly exciting about the issue of crack detection in materials is that it affects pretty much everything around us, the buildings you're sitting in, the chairs you're sitting on. But what was really exciting about my project was getting to design something at the very beginning, go through all the testing, and actually take it through the patenting process to something that might go out into society and have a meaningful impact. And that was truly the most rewarding part, taking research and then conveying it out into the world. Thank you.
And for sharing that, that's much appreciated. I think our judges may need a few more minutes. This is how tough the life of a judge is when lending your time and expertise to the 3-minute thesis competition. So we'll give them a few more minutes. And as we do so, I think we have some more questions coming in the chat box.
Yes, so this question is from Daniel Doherty, and the question was, I'm wondering if and how AI is helping you deepen or broaden your work. So again, if any of the presenters want to answer, if you just want to raise that virtual hand, we'll call you out.
Wow, what a great question. Any takers?
I can go ahead if that's fine.
Yes, go ahead.
Yeah, so with the AI, I think it's really shaping it up, because of what I use the AI for right now. Because before, when I used to do my research, you like deliberate with lab mates and other colleagues, and it really helps you go to lab meetings, but you don't get that many chances to present yourself. So now when a hypothesis or something comes to my mind, I deliberate with the AI, tell it to challenge me if my hypothesis is true or not. There's a problem over here also, because AI also sometimes hallucinates and does something, but I think it's getting better. Some of the platforms like Gemini are getting better at it. So it's good to have a conversation with AI, scientific conversations, which help you understand your own science and probably make it better if there are some good ideas out there. So that's how AI is helping me personally.
Much appreciated. Before we move to Christina, hold that thought. I just want to announce that the People's Choice voting is now closed. So hopefully you got in your vote just in time for your favorite presentation. And on that note, maybe I can loop back to Christina. I believe you wanted to add something about the use, how you leverage AI.
Absolutely, because it's an interesting concept. So if we were to step back and recall, didn't the first models of ChatGPT come out? When was it? 23, 22, around that era? So it's relatively recent how quickly AI is upgrading and being used widespread. So for me personally, of course, I'm a bit hesitant as of right now to truly implicate into the experiments, but what AI is really helpful is for resources, literature searches, and gaining information. So I believe somebody here mentioned that immunology upon first sight was not love at first sight because of how intimidating that is. That is 110% true. And immunology is one of those subjects to where it is not beginner friendly, quote unquote. And there are a lot of receptors, a lot of cytokines, and a lot of different players in this game of protecting your body. So with that being said, AI is really helpful in terms of searching up resources and searching up specific information you want to do in regards to how you want to navigate the system or how you want to prime specific cells to perform specific jobs to protect against specific diseases. So it's very helpful in that regard. Of course, always, and I can't say it enough, always double check the resources. Make sure whatever you're using has the publications and the correct author's years credentials there. But it's definitely helpful in the learning process of going up to par in the field of immunology.
That's a good reminder. It's important to safeguard against AI slop. But I love how you contextualize that for us in terms of the complexity of immunology and all the different players, as you put it, involved in there. So yeah, good comment there. Anyone else wants to chime in on their use of AI or not use of AI, if that's the case? Or else we can move perhaps to another question in the chat box if we have one. Otherwise, I have tons of questions for our participants. I'll pick up on the issue of motivation. We often see in grad schools around the world, and we just came off the Northeastern Association Graduate Schools Conference, which was held here in Montreal. Concordia was hosting it over the last week. It's not abnormal to see motivation dip every now and then during a doctoral or MSc journey, sometimes halfway through as you buckle down and have to confront the blank page syndrome kind of thing, right? Sitting in front of a screen and having to formalize your thought. In all transparency and humility, anyone has struggled with this or has tips they might want to share for jump-starting your writing process, for getting out of that motivational slump a little bit? I know it's a touchy subject, but you've been all just so generous and transparent and genuine. I wonder if you have any thoughts about this. Oh, wow, lots of hands went up.
Ashley, we'll start with you. Ashley Baston.
Thank you. Yeah, motivation can definitely be tough in a PhD program. I think for a lot of people, you go in and you know that you love science, and then you get there and you realize that that is not quite enough to keep you going. And so for me, there have been kind of two arms of keeping that motivation up. And the first one is reminding myself that doing things that I know I like to do, to remind myself that I love what I do. So I really love to present. So you will see me at all of my university's poster presentation days and research days because I love talking to people about my research. And sometimes I need to do something like that to remind myself I'm in this because I really care about this stuff. And even on those really hard days when all of your experiments are failing, you're there because you truly, truly care. And then the other arm of it is kind of reminding yourself that you are still a person outside of the science sphere, outside of the PhD sphere. And so doing things like going for a walk, going to the park, going out for dinner with friends, things that are removed from the school environment to give yourself kind of a chance to reset are so important, because then when you return to those things, you feel more clear and less burnt out. And so it's been, as I'm actually kind of starting to wrap up my PhD, that duality of reminding myself I'm here because I love this and I genuinely care a lot. And also I love myself and I genuinely care about myself. And so to best be the best scientist I can, I need to take care of both of those things. And that has been instrumental in my motivation.
Thank you so much for sharing this so graciously and generously with us. I'll share one of mine, because even as a faculty member or administrator, sometimes your motivation dips as well. And so one trick I found is in my dresser when I get ready in the morning, I've taped maybe 24 proverbs, words of wisdom, things like that. They might be quotes from some famous writer or whatnot that connects me to my why. You know, why am I getting out of bed in the morning? And there's one in particular, I'll share this. I have to watch my language here. We have a coach at Concordia who is famous, Jim Gavin. He teaches in our coaching certificate. He's trained, I think, every coach in Canada. And Jim would work with some of our executive MBA students, and he would put up a slide, and it just read, "Do epic shit." And not all of us, and certainly not every day, we get to do, epic shit. But I've put that on the wall of my dresser in the middle of all the other quotes and proverbs. And sometimes, my eyes will just rest on that as I get dressed, I get ready to come to work, and it's a reminder that, yeah, today's another opportunity to do shit. There you go. So that was my little suggestion. We'll take a couple more comments. I see some other hands up, but our judges are ready to come back with some good news. Before we do so, I do once again want to thank the organizers across the Northeastern Association of Graduate Schools for putting all of this together, putting the resources together, offering the coaching. It's a difficult journey to take on for our participants because it requires a lot of effort, a lot of commitment, but it is supported by wonderful coaches, people who are equally passionate about research communication. So thank you to all of those. And thank you, of course, to the team here at Fort Space for hosting us, doing such a superb job of handling all the tech, all the interfaces, keeping us on track. Yes, big round hand of applause. And of course, the team here at the School of Graduate Studies at Concordia University, Jeff Dover, who had to run to our meeting, Javier, who is behind the scenes, and all the others who came to support our students. We are deeply grateful to all those who support our graduate students. It can be a lonely journey. It can be an isolating one. So anyone who is able to support and add value to the journey of our students, we deeply appreciate it. So before I turn it to our judges, I want to give a chance to our participants who have their hands up to chime in on the question that I ask.
Hannah Wisewell.
Yeah, I mean, I absolutely agree with Ashley. I think that the rivers of your life's blood should come from many different sources, and I'll think that one of those sources is definitely something like this. I was saying this earlier today, that in a competition like this, you have to put your final big impact right at the forefront just to make it engaging for people. And it's nice to be a reminder, because usually when we're working, at least for me, I get tunnel vision where I'm like, okay, I'm working with this piece of aluminum in order to put it on this piece of aluminum. And I kind of forget that I'm really trying to work toward, making aircraft safer. So I think just getting incredibly inspired by events like this can help you remain motivated. So just get out there and start engaging with people besides yourself in the mirror.
I think another trait of our finalists and participants is you're all extremely gracious. A couple of times now, our participants have acknowledged the excellence of others and the level of quality of all the presenters, and I think that's a very gracious thing to do, so thank you for that. We have one last comment, I think, before we wrap it up. One or two last comments.
Chaitanya Parekh?
Yeah, so that's a really good question. So what I always feel, right, and I would tell to all the contestants, all great presentations, and what keeps me motivated is somewhere down the line, this research, what we are doing will matter to someone. It's about incremental progress, which leads to bigger discoveries in future. So that's what keeps me motivated. What I am doing right now might not bear fruit, in terms of impact, but I know somewhere down the line, there'll be someone reading my paper, maybe taking it much forward, and maybe there will be a time then this will lead to a better discovery. So I think with the presentations which I have also seen here, I think that what keeps me motivated, I guess that what keeps everyone motivated, I guess.
Thank you for that comment. And just because we like to keep the suspense going just a little while longer, we'll wrap it up with Zahava's comment.
I'm so sorry. I'm going to keep this so brief. I just wanted to say that When I get into those slumps, for me personally, that imposter syndrome really just comes out full force. And I think what helps me and what I think is really important is that everybody goes through those moments. And so a lot of people put their best foot forward, and kind of what you see, similar to social media, is their best, but internally they're going through very similar struggles. So leaning on your colleagues, leaning on the people you think are doing such a great job, and knowing that everyone's going through the kind of the same journey at different points is really important.
Thank you. Thank you for reminding us also of the sense of community, that you are not alone, even though it's a bit of an individual journey, an isolating one. You are not alone. You have friends, you have colleagues, you have mentors, you have supporters in your corner. We now come to the moment that we've all been waiting for. But before we announce the winners and I invite the judges to present the awards, I want once again to congratulate all of the finalists, all of you. Please, a big round of applause. You've made it, you've done it, you've shown once again the high level at which you can rise, and you've managed to convey your research in a compelling, easy to understand manner. So I hope that tonight you can take a moment and savor that victory, savor the moment, and give yourself a big pat on the back. So I'll pass it over now to the judges who will present the awards.
Hello, everybody. My name is Dr. Yvonne Lee. I direct graduate writing support at Lehigh University in Eastern Pennsylvania. And it has been my pleasure to be here and listen to all of your amazing presentations and the research that you're doing. It's part of the reason I love my job. And you know what? I wasn't going to say this, but I would like to piggyback off of Zahava specifically and say, use the community around you. Join a writing group, go to a writing retreat, form a writing group or just a working group, and you'll find other people are feeling the same way that you're feeling. That's part of the job that I do as well. All right, so one more thing. Before I announce the People's Choice winner, I just want to say thank you to all of you for all of the heart, knowledge, and time that you put into the research that you do, and for sharing that enthusiasm with us today. It's been amazing to watch. All right, so are you ready? The People's Choice Award is going to Mariam Afkari. Congratulations! All right, I will now pass over the mic, so to speak, to Giovanna to announce our first co-winner.
Thank you so much, Yvonne, and congrats to Mariam for winning the People's Choice Award. My name is Dr. Giovanna Milosali-Cedean. I'm a graduate student success director at the University of New Hampshire. I'm also the lead of the 3-minute thesis. I'm so grateful for all of you guys, for presenters, for the work that you do. As a past winner of 3-minute thesis, I know how much work this takes, and I admire you all for taking the time on top of all your graduate studies and everything else that you do. This is a lot of work, and you've done an amazing job. You're already winners. You've heard that, but I hope you really know that. I also really want to make sure that we thank the organizers. Thank you so much to Jeff, to Jordan. Jordan, you did an amazing job emceeing. Javier, big shout out to you for managing this big event. We heard at the beginning it's 46 institutions that participated, so a heavy lift to organize all of that. A huge thanks to my colleagues, Yvonne and Amy, for judging. It was really a pleasure. It was such a fun task, really hard, but really fun task to do. Many thanks to all the audience and everybody who supported our presenters. They did the hard work, but help of supporters really is important. So we have 2 co-winners. I get to announce one of them, and then I'll pass it to Amy for the second one. Big congratulations to Hannah Wiswell from Princeton University as one of the 2 co-winners. Great job, Hannah. And now passing on to Amy for the second co-winner.
So, good afternoon, everyone. I'm Dr. Amy Sisson. I am the AVP and Dean of Admission at Wentworth Institute of Technology in Boston. It has been really inspiring to spend the afternoon with all of you. It's really incredible. It was also incredibly difficult to make these decisions because everyone is just excellent. Again, you are all winners. But it is my distinct pleasure to present the second co-winner to Christina Stalling. Congratulations.
Let's go!
Thank you so much. Thank you.
Well, congratulations to Hannah, Christina, and Miriam. And again, to all the finalists, a big, big heartfelt thank you to you, Amy, Yvonne, and Giovanna, for your hard work. It is indeed hard work to keep up with all those presentations, the fast pace, and to bring discernment and judging this. It's really a tough job. So congratulations again to our winners, to all the presenters, the judges, the guests who joined us here and online. On behalf of Concordia University and the School of Graduate Studies, thank you for being part of the 2026 Northeastern Association of Graduate Schools 3-minute thesis regional competition. We hope that you leave today inspired by the quality, the diversity, the impact, the hope that the research that was presented allows us to have in the fate of humanity and the future of our planet, but also that you take away how important words are, not just in a 3-minute presentation, but every day. We can all use our words to inspire, to comfort, to heal, to forge better ties and relationships. So as you go forward, ask yourself how you might do the same and honor the impact and the legacy that our finalists have shared with us today. So thank you so much, and have a wonderful rest of your afternoon. Thank you.
Thank you, Jordan. Thank you, everyone. I just wanted to thank the people joining us online, our audience as well there, and those of you here in the space, those of you on YouTube. We're going to close up our Zoom and our live stream now, but this does remain available on our YouTube channel, and obviously you're going to revisit it and share it. Have a wonderful afternoon. Thanks again, everyone.